Welcome to Cancer HealthCast, where science is driving hope. I'm your host, Nikki Henderson. Today, we are joined by Dr. Henry Rodriguez, who is the founding director of the Office of Cancer Clinical Proteomics Research at the National Cancer Institute. He'll be talking about two major NCI programs that are helping to improve clinical data sharing. He will also discuss how NCI is incorporating AI and what the future holds for precision medicine. Hello, Henry, and thank you so much for joining us on the show.
Hey, Nikki, thanks for having me.
Well, let's go ahead and dive into our discussion. So, Henry, to start off, I want to learn more about your career up to this point. Could you elaborate on how you got to your current role as director of the Office of Cancer Clinical Proteomics Research, and tell me about your time at the White House?
Yeah, sure, Nikki. Well, so, it's interesting because that's sort of a journey so let me kind of give the overall picture here. So my journey to where I currently now stand at the National Cancer Institute, in a way, it's a good example on how to seize opportunities when they're presented by life, because in many instances, they could actually lead to really big rewards. So first and foremost, I'm a Cuban American. I'm born and raised in Miami, Florida, where both of my parents immigrated to the United States. Now, in high school, that's really where I got interested in science, especially in the health sciences. However, one of the things when I got into college, first I went to a community college and later I went to its feeder university, that I quickly realized is that blood, especially moving blood, makes me uncomfortable. So I decided to focus on research instead. Now, I'm one of those classic ones. I studied biology, I minored in chemistry, all the way through college and in grad school, and ultimately I specialized in cell and molecular biology. Now, my interest in cancer research, which kind of has led me to the path where I'm now, really began to take hold when my mother actually battled breast cancer. And this is when I was in my initial postdoc fellowship at the Scripps Research Institute in La Jolla, California. Now, fortunately, she's doing well. She's a long-term survivor. But that was really a turning point for me. So I quickly decided then to pivot. I now moved to Los Angeles. I trained at the City Hope Cancer Center. And the reason I went there is because I wanted to learn about that transition that exists between research that I was doing and clinical implementation, which is the ultimate goal that we want to do towards the latter end. Now, initially, I planned to go directly in the industry. I mean, that was my focus because I really understood that model. But what happened to me was I was at a conference in San Diego, and I was approached by an individual that when they gave me their business card, it says the National Institute of Standards and Technology, NIST. And they apparently were looking for an individual to join a new program that they were sort of kicking off that focused on DNA technologies. Now, even though, I have never heard of this institute, let alone when I tried to look it up, why this institute would even exist within the Department of Commerce. I kind of was intrigued. I ultimately visit the institute, which is just outside of Washington, D.C. I went to learn about its mission and its plan to build these programs that they wanted to build in this emerging field of biotechnology at the time, also with the bioeconomy. And I kind of saw it as a learning opportunity. And quite frankly, it was, and it paid off. So at NIST, I actually held several roles. Ultimately, I became a founding director of one of the research groups. And there, we started to dabble in genomic-based sciences, proteomic-based sciences, imaging, and biomedical databases. And I also was really neat because I also got to serve as an analyst in the office of the director, where then I learned to coordinate and implement several technology programs across the entire institute. But what happened then, the National Cancer Institute, I think now is around 2006, I kind of put a calendar on this. They were actually looking for an individual to lead a brand new effort that they wanted to kick off that focused on proteomics, large scale. And this was sort of a tough decision for me because I was kind of growing in my career at NIST. So it's risky, but at the same time, I thought it would be a really great opportunity to be able to learn and grow I mean, let's face it, at the federal government's principal agency, right, for cancer research and training. And it was an opportunity that also, for selfish reasons, would bring me back into a clinical research environment, which I was starting to miss because that's what I ended up doing in California. Looking back, I'm glad I did it. I mean, because it's really given me the opportunity to work and grow with some of the most talented people across federal agencies, academia, and industry. Now, one of those major highlights that you just alluded to, it's kind of like a capstone moment in my career, which was being called to work at the White House. So firstly, in 2016, this is when then Vice President Biden, he has to call for the cancer moonshot. And when that went public, it really rallied many of us within the research community. I mean, I was sort of galvanized with it and its overarching goals. Now, I got involved with it when the White House Cancer Moonshot Task Force colleagues there, they asked me for ideas and would I be interested in leading an effort that might begin to bring together research centers around the world. Now, my focus, my twist that I put into it was, well, maybe doing an area of research that the office that I lead is already helping to pioneer. And that experience was really, truly impressive. And it sort of reached a peak for me at a gala event that we hosted, where then I had even the opportunity to sit alongside then Vice President Biden throughout the entire event. Now, in 2021, now we're under the Biden-Harris administration. And there I got asked ultimately now to serve as the Assistant Director for Strategic Health and Cancer Sciences in the Executive Office of the President, specifically within the Office of Science and Technology Policy. I mean, working on initiatives like reigniting the U.S. Cancer Moonshot, collaborating with colleagues in Europe with their, with Europe's Beating Cancer plan. I mean, this is beyond incredible for me. This kid from Miami, Florida, to be in this space with all these different parts across the U.S. and now here in Washington, D.C. So ultimately, quite frankly, seeing science at the policy level or policymaking level, ultimately for me was truly a dream come true.
Wow, Henry, that was an amazing story and an amazing journey. And also, it's so good to hear that your mother is doing better as well. Thank you so much for sharing. Well, what is NCI's Office of Cancer Clinical Proteomics Research? And could you define the concept of proteogenomics?
Yeah. So Office has a very long words behind it. So like a lot of stuff that we do here, we will put acronyms, OCCPR. But so the office from an organizational perspective, so we're part of the Division of Cancer Treatment and Diagnosis. Specifically, we're under the Office of the Director in that division. Now, our main goal is to use proteomic and proteogenomic science approaches. Ultimately, what we want to do is make cancer treatments and diagnosis better. Now, another goal or mandate, really mission of our office is to create public resources. And these resources are things such as multimodal tumor molecular characterization maps or these very large complex data sets. We also develop, for example, fit-for-purpose targeted proteomic assays, and we also develop reagents, such as very well-characterized antibodies. And these are given back freely to the public as a way to help speed the process of turning scientific discoveries that other people are doing, potentially into practical treatment for patients. Now, in terms of defining that, that spin-on terminology is what now we call proteogenomics. Let me kind of put it into simple terms. So genomics, that a lot of people are going to be very familiar with, that looks at the genetic instructions that gets provided by our DNA. And subsequently, that gets transcribed through the RNA. But this is what many of us, almost everyone, is going to refer to as our molecular blueprint. Now, proteomics, what that does, that actually looks at the proteins that gets translated from that blueprint. Right, so DNA, but really from that RNA. This is people that are in the biology space, what we refer to as the functional environment of biology. I mean, proteins are the workhorses in our cells. And it's not surprisingly that proteins is what most drugs will actually target. So what we do within our office versus treating each one of these scientific disciplines in isolation, we basically merge them together. So proteogenomics is the combination of both of those approaches, being able to conduct comprehensive genomic-based measurements and protein-based measurements to get a real picture, a complete picture when we profile a tumor from a patient.
Okay, I see. Well, Henry, what are some of the key objectives your office is trying to accomplish when it comes to detecting, diagnosing, and treating cancer?
Hmm. So the main objective of our office, I would say, is ultimately to systematically identify the proteins that arise from changes in cancer genomes and related biological processes. Now, and what this does ultimately is that it helps it helps us actually to go deeper into cancer at that molecular level, especially areas where genomics alone cannot fully explain the biology. Now, our ultimate goal is to basically speed up, that process, the translation of molecular discoveries into practical clinical applications. Now, to achieve it, the way that we do it is the programs that run out of our office, they then perform that molecular characterization of a patient's cancer in that integrated approach. So for every tumor, again, it undergoes sequencing of the DNA, RNA, protein-based measurements, what we call proteogenomics. We also combine it with images from the pathology suite, images from the radiological side. And with this approach, what we do is we help to, for example, refine, prioritize driver genes. We help to gain new insights into cancer subtypes. We help to understand dynamic changes that exist within cancer signaling pathways. And we also help to, for example, enhance our understanding on how cancers react to and unfortunately might resist therapies that are being studied.
Perfect. Well, I know your office oversees NCI's flagship CPTAC program, the CPTAC program, as well as the ICPC initiative. Could you quickly define these programs for me?
I think it's good to know the origin on how, especially CPTAC got created, because that really puts things into context. So let me kind of first give a little brief overview of the history from my little, myopic way of viewing precision medicine. So about 25 years ago, around the year 2000, that's when the first draft of that Human Genome Project gets released. And that's a huge, momentous achievement. I mean, that's this off this chain of events, right? I mean, people start using those terminologies. Medicine is going to now become very precise, and claims are being made within a window of time that we're going to transform the way that we identify and treat individuals with the many diseases of cancer. Now, to the credit of NCI, about 5 years later, around 2005, one of our advisory boards, the National Cancer Advisory Board, they start asking, well, seeing where technology is today and our ability to characterize samples at that level of molecular information, what can we do in the world of cancer research with it? So the National Cancer Advisory Board, what they do is they actually spun out this working group to focus on those biomedical technologies. And they ultimately came back and they gave their recommendations to NCI to take into consideration. And in there, they had things such as, which is what all, we're always trying to do. They talked about restructuring the clinical trials to make them more nimble, to make them smaller, to make them smarter. But one of the other things that they did touch upon is they recognized the opportunity that now is at hand to be able to take cancer and to learn it at that level of molecular information would be very powerful. So they suggested you should build programs in genomics, but they also took to heart what we all learn in biochemistry, DNA goes to RNA and goes to the protein. They also recommended to build a program that would complement the genomics at that protein space. At the same time, they also understood that most likely this is going to be team-based programs. You need to develop ways of this voluminous information of data that's going to be generated. How do you share the data amongst the labs? And more importantly, how do you give the data back to the public? Now, NCI, to its credit, started to address these things just about a year later. So in 2006, that's when they created the Cancer Genome Atlas, TCGA. They created our program, CPTAC, the Clinical Proteomic Tumor Analysis Consortium. And now more recently, NCI, we've now developed ways for the public, the public at large, to get access to this rich information. And that's through the many data nodes that we have within this ecosystem that we refer to as the Cancer Research Data Commons. Now let me pivot a little bit back here. I talked about that Human Genome Project. What that did was it gave a lot of confidence to the public in the sense that genomic-based technologies, they believed that were highly reliable and highly reproducible. Now what happens around that window of 2005-ish, midstream, before NCI launches, these flagship initiatives a year later, a publication gets released. And the publication strictly looks at proteomics, and it focuses on early stage ovarian cancer. And it had these claims, but unfortunately, subsequently, the public, it was found that there were numerous errors within the way that study interpreted the information, the way it designed the study. And it sort of put a damper on the idea of going full throttle with genomic characterization to figure out the biology and full throttle at the protein characterization to figure out the biology, because they had reservations, to put it in a mild way, at the protein side. So CPTAC, its original origin, to the credit of NCI, I get hired in 2006, and they decided not to throw away the baby with the bathwater. TCGA goes full forward when it comes to the genomic characterization of tumors. CPTAC, at that point, its mandate is figure out the analytics. Take these powerful platforms and determine the best you can whether you could generate rigor and reproducibility with them. So it took 5 years, but at the end of the day, we demonstrated that. So now we go back to the leadership of NCI and back to the extramural communities. And the next iteration is, let's revisit back what the Cancer Advisory Board suggested. So now we collaborate with the Cancer Genome Atlas. We obtained samples that already went that comprehensive genomic characterization. And now we were characterize it at the protein level. And it was a simple thing that was being asked. Can you identify additional biology? That's it, additional biology that was not identified through that genomic sequencing that was already done. And to our credit, we did for all those 3 cancer types, and biology that simply is not discernible through genomic-based sequencing. So the current iteration today of CPTAC, it's a US-based operation. It is funded what we refer to as extramural programs, academic laboratories. And what they're involved in, these leading centers nationwide, they will perform that comprehensive proteogenomic characterization of tumors. And all that information gets released back to the public. At the same time, we also support now clinically relevant research project. I think one of the things that's quite nice about CPTAC is that it has now enabled NCI and the rest of the world, to gain access to some of the largest curated public repositories that exist of proteogenomics data sets. And you can actually, quickly gain access to this through NCI's Proteomic Data Commons, which is one of the nodes within the Cancer Research Data Commons. Now, the international one, that ICPC that you referred to, that one was actually created in 2016. That's the one that I referred to, that in 2016, when the first cancer moonshot went public, that I was asked to sort of build this international activity. And the impetus was kind of straightforward, right? Can we create a mechanism that would encourage the use of standards among laboratories, and not just within the US, but now amongst various countries to ensure rigor and reproducibility, because you want that assurance that the measurement that you obtain is not an artifact, whether it be an analytical or a computational, or even the way you collected these materials, right? You want that measurement to be representative of biology. And at the same time, we simply wanted to promote collaboration, coordination, and data sharing in proteogenomics research among research institutions worldwide. In 2016, we kicked that off, and the very first meeting was held in Taiwan because the Human Proteome Organization was having a conference there. And I think at the time we only had like 7 countries on board. Today, that little program now spans about 20 research sites and involves 14 countries. And I think one of the real highlights of that is in 2017, just a year into the launch of this whole idea of this international activity, is again, the Human Proteome Organization was hosting a conference in Dublin, Ireland. And the different investigators from all these laboratories at that time decided to get together for a function. And I reached out to people who knew then Vice President Biden. You were the ultimate cheerleader. You're the reason that galvanized, not just within the US, but all of us within this international effort to create this activity. Would you like to attend? And then that's how he ended up attending that gala. And it was, again, it's serendipitously what you don't think is going to happen happens. And when it happens, it's actually quite nice.
That is incredible. Oh, my goodness. Well, how do these programs that you just mentioned enhance the sharing of research data as well as accelerate cancer research and improve patient care? And could you also talk a little bit about why the international aspect of these programs is so important?
So as I indicated, so one of the objectives of our office, any program that we create, is ultimately to develop public resources, and data sharing happens to be a very key component.
Mhm.
I mean, I personally believe, I'm convinced that within the scientific community, there's an understanding that sharing research data allows those resources, right, that a lab will generate, allows those resources to have a much broader impact beyond the initial analyses that's able to be performed by that one lab. I mean, besides confirming original findings of a study of other groups, which is important to do, I mean, raw data could be utilized to investigate new hypotheses that other investigators are now performing, especially when you want to integrate that, data that you generate with other publicly available data sets. It just increases your power. Now, in terms of a global perspective, yeah, that's a good one. I think personally that any objectives that brings together researchers across the globe as a way to streamline your efforts, you solve problems together, you exchange your research data, you're going to be in a really good position. You're going to be well suited to tackle the challenges that faces precision medicine. For instance, let's just consider rare cancers. So here, just being able to obtain sufficient specimen patients for studies is already a daunting exercise, right? So that's difficult. So collaborations, especially and with sharing that data, is now important for simply pulling resources and expertise in order to advance research within that space. Another one is if you study cancer, the differences worldwide, and sharing the data now that becomes representative of different geographical regions, that really begins to help us now to even better understand cancer. And hopefully it could lead to treatments that work not just for one pocket of individuals, but for more individuals. The bottom line is that research that crosses, for me, international borders, I think that's a fundamental building block of precision medicine. And the wider our reach, the greater the number of patients, and that's going to be whether at home and abroad, that ultimately is going to benefit.
Okay, great. Well, we're going to shift gears a little bit here. What role does artificial intelligence and machine learning play in helping researchers better understand proteogenomic complexity of tumors?
Yeah, that's a great question. I mean, first of all, this is a space that has a lot of unknowns. However, personally, I see whether it be AI, ML technologies becoming an ever more important tool when it comes to multi-omic analysis, such as proteogenomics, right? Especially in our context, where we couple a tumor's molecular characterization data, so it's DNA, RNA, protein-based sequencing, also with its imaging data, so H&E images that, again, we get it from the pathology suite, DICOM images that we get that from the radiology suite. I mean, the reason it's becoming important is because those new tools, from what we could see, is able now to begin to provide speed, accuracy, sophistication, to be able to deal with simply the voluminous, diverse, high-dimensional data sets that is now becoming standard in a lot of these activities that now we perform. I mean, I personally think that combining the exponential growth of multi-omics data, whether it be with artificial intelligence, machine learning, or even deep learning, I think that has a really good potential to make a great impact. For example, in cancer subtyping, risk stratification, prognostication, prediction, and even potentially clinical decision making down the road.
Okay, great. Well, Henry, what direction do you see precision medicine heading in the future? And also, are there any future initiatives that you're excited to talk about?
Yeah, so for that, I have to break out my crystal ball, right? So, so, so for me, Nikki, I mean, I think that in order for precision medicine to continue to evolve and grow, this is me speaking. I think ultimately it needs to move beyond sequencing. So, so colleagues and I, we actually wrote a perspective in the journal Cell, I believe in 2021-ish. And I think that was called Next Horizon in Precision Oncology. So in there, we talked about multi-omic based approaches, bringing that now towards clinical trials. We talked about the importance of data sharing, and we also sort of tiptoed around artificial intelligence. But but let me be clear. I mean, today we know we know unequivocally that cancer is ultimately it's a disease of the genome, and this is now evident through the major strides that have been made using genomics as a way to understand and treat numerous diseases that's associated with cancer. And you could really begin to even quantify the impact. Like one of the metrics that you could look at, for example, is cancer mortality rates in the United States, especially when you drill down into data. One of them is lung cancer, specifically non-small cell lung cancer, where now we target EGFR in first-line therapy. However, I believe that while cancer experts predominantly use genomics as a way to understand tumors' molecular characteristics, I think it only paints part of a picture, which in a way is a restricted view of a tumor's underlying biology based on what we've been discussing today. Furthermore, I think that despite scientific advances, there's also non-scientific obstacles that we still need to overcome. I mean, cancer is a global issue that affects many. And unfortunately, still too many lives are being lost to its many diseases. So, I mean, for me, I would say a scientific priority would be to include proteomics alongside genomics. So this is the proteogenomics in guiding personalized treatment plans for cancer patients, right? So like that's a dream. And I think with the wealth of information that you find, that you get now in proteogenomic data, I believe that we will soon be reaching a point where we can make this a reality more and more. Now, on the non-scientific side, there's still challenges. And some of the challenges are, for example, scale of certain types of research. So some are very costly. Issues, if you're talking the private side, issues with reimbursement when it comes to diagnostics. And quite frankly, there's still barriers that remain in our ability to share data. This is just to name a few. Now, my ultimate dream in terms of future initiatives, sort of the evolution, I would say, of CPTAC, I could kind of view it as, one could phrase it as next generation solutions. And to me, those solutions would be based on kind of what we've been alluding to, algorithmically guided patient care decisions. And those decisions would come from, for example, AI-driven multi-omics, proteogenomics, clinical, and longitudinal studies. And of course, involving the sharing of research data. The true success for me, when I could say we're done, we've reached the top of the mountain, is when precision medicine, we could simply call it medicine.
I see. Well, Henry, this has been just a wonderful conversation, and I really appreciate you sharing just some really valuable information with us. So I want to thank you for taking out time to speak with me, and I hope that you have a great afternoon.
Thank you, Nikki.
HealthCast, along with GovCast and Cybercast, is a production of GovCIO Media and Research. To explore our content, visit our website, govciomedia.com. Keep an eye out for new episodes every Tuesday. And if you like what you heard, leave us a review on the podcast platform of your choice. Have a topic you want us to discuss? Contact us at newsletter@govcio.com.